First of all, I would like to thank you everyone for joining this webinar. In this session, we will have a discussion about bispecific antibody discovery using an integrated platform at GenScript ProBio. Let's start with a quick glance of the market trending for therapeutic antibodies. Over the last decades, monoclonal antibody therapy has experienced an explosive growth. However, given the limitation of monotherapy and druggable targets, more and more people realize that bispecific antibody could be a shortcut to best-in-class or first-in-class. As a result, bispecific antibody is expected to achieve a much higher growth rates in the next few years and take a fair share of the market in the next decade. Currently, there are almost 1,000 bispecific antibody pipelines in the preclinical and clinical stage. So far, 2 products from Roche and Amgen has been successfully launched, making more than $3 billion every year. Among these 1,000 pipelines, more than 3/4 are still in preclinical stage. For those in the clinical trial, more than 60% are T-cell engager, which is the earliest concept of bispecific antibodies. However, other MOA involving dual targets inhibitions and dual epitopes are quickly catching up. Comparing to monotherapy, bispecific antibodies and the combo therapies has demonstrated its great potential to achieve better efficacy and avoid drug tolerance. Although combo therapy offers a better flexibility in terms of dosage, combination of targets, bispecific antibodies is expected to be a better choice when it comes to safety and cost. In addition, bispecific antibodies is showing superior potencies in certain MOAs, such as T-cell engaging by redirecting the T-cell activation in the killings, modulation of the receptor signaling, simultaneous activation or co-inhibitory receptor or checkpoints, and targeting multiple epitopes on a same target for improved efficacy. Next, we will further discuss the rationale behind the format designing bispecific antibodies. Currently, there are several dozens of different formats for bispecific antibodies, the majority of which are IgG-like structures. However, most of this designing involves mutations and the reformattings. For instance, knob in whole is one of the earliest concepts to force the heterodimer pairing of heavy chains. CrossMap took a step further to switch the CH1 and the kappa chains to reduce the mispairing of the light chains. A different approach to avoid mispairing issue is to reformat one of the IgG to single chain F3 and fuse it to the other IgG molecules. These engineering works usually creates new issues down the road. First of all, complicated structure leads to unpredictable immunogenicity risk, adding more difficulty to the already challenging work. More notably, people will experience all kinds of developability issues with most of the bispecific antibodies, such as unwanted homodimers, aggregations, solubilities, And most of these features eventually lead to manufacture problems, including low yields, low titers, complicated purification process, and etc. In order to overcome these issues, GenScript ProBio offers a SMAP body bispecific antibodies, which is built on IgG fusing with VHH single domain to either terminus of heavy chain or light chains. This creates a fully symmetrical structure without any FC engineering or reformattings. Both IgG and single domains are naturally generated, making it more predictable in terms of immunogenicity. Moreover, this unique structure gives the antibody exceptional developability and manufacturabilities. It behaves almost identical to traditional IgG, but with much better flexibilities in terms of targeting multiple epitopes, multiple targets. In addition, multiple single domains can be fused onto the IgG at different locations with different copy numbers to accommodate the MOA and to achieve desired efficacies. To better demonstrate the correlation between bispecific antibody formats and its effect on antibody activity, I am showing a direct comparison of 3 different types of bispecific antibodies. The first one is represented by knob-in-hole and cross-map. In this set of experiments, we use the sequence of avelumab and IMAB 362 to evaluate the activity against PD-L1 and Claudin 18.2. Judging by the dose curve of the cell-based assay, both formats severely compromised the efficacy of each parental antibodies from 20 to 100 folds, as measured by the PD-1, PD-L1 blockade assay to the left, and the Claudin 18.2 ADCC assay to the right panels. There could be several different reasons for this decreasing activities. First of all, the reformatting from IgG to cross-map reduce the valencies. Basically, a typical IgG is bivalent. There are 2 antigen binding sites in each one molecule of the IgG. However, when it switched to cross-map, there's only 1 binding pocket for each target on each arm of the antibodies. So this could be one of the reasons explaining the decreasing activities. Another possible reason could be the engineering, including on the FC part, the knob and the holes, and also the switch between the CH1 and the kappa. These parts of engineering could have some negative effects on the antigen binding sites, leading to the decreasing affinity, decreasing activities. In the second set of the studies, we choose the format of IgG fused with single-chain FV. And the same sequence of avelumab and IMAB 362 were used in this case studies. Unlike the first group, this format is able to retain the activity from the IgG parts. However, the SCFV reformatting also significantly impacted the efficacy of the parental antibodies, most likely due to the loss of affinities. So as you may well know, the reformatting from IgG to single-chain FV always lead to the decreasing affinity from 10 to 100, even 1,000 folds. And this decreasing affinities may explain this decreasing activities. In the third group, we adopted the SMAP-body format using the sequence of IMAP 362 and invafolimab to build the bispecific antibodies. Just like what we expected, this particular format is able to retain most of the activities from each parental antibodies based on the data from the PD-1, PD-L1 blockade assay on the left panel and the ADCC assays on the right panels. In the next part, we will share an antibody discovery case study of PD-1 and the TIGIT bispecific antibodies using the SMAP-body format. So this is a general workflow of therapeutic antibody discovery. In the lead generation stage, multiple platforms can be utilized to screen for early stage candidates. Hybridoma and phage displays are well-established technology and has been proved as a reliable approach. In addition, single B-cell screening is another rising technology, which is building track record really fast, especially in infectious disease field. Upon obtaining initial candidates, cell-based functional assays is a common tool to evaluate the biological efficacies in earlier stage as a much cheaper substitute for expensive animal models. Depending on the origin and the desired feature of the antibodies, functional leads will go through antibody engineering, such as humanizations, affinity maturations. At this stage, usually the total number of candidates will be narrowed down to less than 5, and the final ranking will be determined through developability assessments and animal model in vivo efficacies. So back to this case study, we use both the PD-1 and the TIGITs are inhibitory checkpoints. So our purpose is by targeting these two inhibitory targets, we're hoping to synergistically to restore the T-cell activities. So after obtaining functional lead of anti-PD-1 IgG and anti-TIGIT single domain using traditional hybridoma and immunized phage display library approach, one top candidates from each target, so one from PD-1 and one from the TIGIT, were selected to construct the SMAP-body bispecific antibodies, the BTP11 and BTP13. First of all, we evaluated the binding affinity towards the cell surface expressed antigen using FACS. So both bispecific antibodies not only retain the comparable affinity against human antigens as its parental monoclonal antibodies, but also shows a decent cross-binding to cyno and mouse target antigens. In addition to FACS, the SPR Biocore assays is also used using recombinant proteins, also demonstrate cross-species binding to cyno monkey and mouse. The difference of kinetics is within the range of 2 to 100-fold, and the minimum affinity is at the level of 10 to the -8, which makes it feasible to translate the data of animal study to humans and avoid the need of generating surrogate antibodies for efficacy and safety studies. Besides the binding affinity data, we also characterize these bispecific antibodies through functionalities. In this FACS-based receptor ligand blocking assays, both bispecific antibodies are able to disrupt the interaction between target and its ligands. So in the left panel, which is the PD-1 and the PD-L1, in the right panel is the TIGIT and the CD155. And this blocking capabilities is also comparable to their parental monoclonal antibodies. Moreover, cell-based function assays also demonstrate that both bispecific antibodies, the BTP11 and the BTP13, are able to block the PD-1, PD-L1 signaling and the TIGIT CD155 signaling, and also activate the T cells by the measurement of induced interleukin-2 to a similar level as their parental antibodies. To further characterize the dual antigen targeting profile of these two bispecific antibodies, we conducted a SPR vehicle-based binding assays in which the human or the cyno monkey TIGIT was immobilized onto the chip to capture the bispecific antibodies, followed by the titration of human or cyno monkey PD-1s. So this kinetics data actually proves that both BTP11 and the 13 can bind to PD-1 and the TIGIT proteins simultaneously. Besides antigen binding, we also established a bifunctional cell-based assay to further evaluate the dual antigen blocking. So in this model, the reporter gene is induced only when both PD-1, PD-L1, and the TIGIT CD155 signaling pathway are inhibited. The results showed only the bispecific antibodies BTP11, but neither of the monoclonal antibodies is able to induce the signaling. Moreover, the same bifunctional cell-based assays was utilized to further compare the efficacies of these bispecific antibodies versus the combos of two monoclonal antibodies. And the results demonstrate that the BTP11, the bispecific antibodies, showed a similar or even better activity in either equal weight concentration to the left panels or in the equal molar concentration to the right panels. In addition to all of those in vitro data I have shared in the previous slides, we also evaluate the efficacy of the bispecific antibodies BTP11 using a human PD-1 knocking CT26 CDX mice models. 4 groups of mice were treated with either irrelevant IgGs, the anti-PD-1 IgGs, the bispecific antibody BTP11s, and the combo of both monoclonal antibody IgG and the VHH, respectively. 3 treatment groups showed significant anti-tumor efficacies, but the bispecific antibody groups gives the best rate of partial response, which is 3 out of 5 as compared to 1 out of 5 for the combo and 0 out of 5 for the mono IgG treatment. To further confirm the in vivo efficacies, we did the similar tests in a different model, the MC38 CDX model, using human PD-1 knocking transgenic mice and obtain a slightly different result. Both irrelevant IgG and the anti-TIGIT VHH, the single domain, didn't show much anti-tumor efficacy, as showing here with the color black and blue. While the bispecific antibodies, the BTP11, the reds, the IgG monotherapies, the green one, and the IgG VHH combo, which is the purple one, gave similar tumor inhibition rates. And there is not much difference in terms of the body weights along the whole course of treatment for all 5 groups. Besides biological activity, developability is probably the one with most concerns. We did a preliminary developability evaluation to assess the thermal stabilities, expression levels, solubilities, cross-reactivities, and the stabilities under stress, like repeated freeze-thaw cycles, acidic or basic conditions. So this particular bispecific antibodies, the BTP11, showed comparable characteristics in all of these indicators as typical IgGs. After all of these characterizations in those previous slides, we decided to choose the BTP11 to generate the stable cell lines. And 5 single cell clones were obtained, and all of them showed a 2.4 to 3 gram per liter titers using fed batch. So at the end, a quick summary of what we have talked about. First of all, GenScript SMARTbody is one of the most natural bispecific antibodies with impressive developabilities. Comparing to monoclonal and other formats of the bispecific antibodies, it demonstrates better activity and efficacies. Plus, this is an open access platform available for different business models, including fee-for-service or co-developments. Before I conclude this session, I would like to give a few words about ourselves. GenScript ProBio is now offering a one-stop solution for both therapeutic antibodies and gene cell therapies, including early-stage discoveries, preclinical developments, manufacturing for clinical and commercialization stage. In the discovery field, which happened to be my area of expertise, we provide services from target discoveries all the way to PCC. We offer a variety of tools, including hybridomas, phage displays, single B-cell screening, and fully human transgenic rodent for early stage discovery in different modalities and different therapies. We have been providing discovery services for almost 2 decades, and at least a dozen projects previously delivered are in clinical trials. And at least 1 product is launched in Asian Pacific area. I believe our experience and the technical expertise is going to make a difference to accelerate your drug development business. So that's all for me. Let's move on to the Q&A session.